All right, so hitting a turning point today, I guess you could call it. We're finishing up talking about the kind of microscopic level content of the course, building from individual atoms, now finishing up the last section that deals with molecular biology. Um, but don't let that get too stressful. Just going to talk a little bit about DNA today and focusing on its structure and how it actually is able to make copies of itself. As I've said before, um, if you were in a full-blown biology major, you'd need to know a lot more detail about photosynthesis and cellular respiration and a lot of other things like DNA and how it works and how it makes copies of itself. But for the purpose of this class, again, I'm just looking for you to have at least a basic level of understanding of what it looks like and a basic understanding of how it actually makes copies of itself. After today, we're going to skip ahead, and then if you've looked ahead at all in the book, um, we're jumping to chapter 10. I mean chapter 11. We're skipping chapter 10. It's an interesting chapter about biotechnology, and certainly, you're welcome to read through there. It's relevant, to the world today, of course, and even more on into the future. But again, we only have so much time in the course to cover material. And then from there, we're going to chapter 11, 12, and then we skip ahead to 20, go back to 19, and then finish up the semester with chapter 21. So there are a bunch of chapters in the middle of the book that deal specifically with things like how the body works, going through talking about, all the different systems of the body, muscle, skeleton, etc. And again, while that is really interesting stuff, and if you're trying to understand life in general, then understanding how it works is pretty important. But again, it's kind of beyond the scope of this class. If you're interested in that, we offer other courses on campus that are meant for nursing, pre-med, that kind of stuff. But they're certainly open to anyone who's interested and is willing to work hard to, study the material to learn what's going on. And so as we move into chapter 11, 12, and beyond, we're moving from the stuff you can't see to the stuff that in some cases is almost too big to see. The topic in chapter 11 is evolution, which is a process that occurs over long periods of time. And typically humans, we have a pretty limited perspective on time. We can't really think or see millions of years. If we're lucky, we get 100 maybe. And then other processes about diversity and more general ecology and some other interesting stuff to kind of wrap up the semester. But before we get there, back to DNA. We've actually had huge advancements in DNA going back to the 50s when we first, meaning science, first discovered the structure of DNA. As we talked about with Mendel, he didn't know DNA existed or genes. He just hypothesized that those things must be there for him to be able to explain what he observed experimentally. Since then, and particularly since the 90s, we've made huge strides in our knowledge with respect to DNA. A lot of it we tend to think about with things like crime scene investigation, being able to go from the old days where even 100 years ago to try and figure out who might have committed a crime. Maybe you got lucky and found a fingerprint, and then maybe you got really lucky and happened to have a suspect whose fingerprints you took, and then we're able to match. Even today, we've got databases that you can scan and find fingerprints. Back not that long ago, but 40, 50, 60 years ago, all we had were, if you actually had a card with somebody's fingerprint on it and you had the time to look at every one of them and got lucky and found the person who committed the crime. But now we have the ability, even with a single cell, a single little skin flake or a hair follicle, that we have technology like PCR, polymerase chain reaction, where we can take that tiny little sample and magnify it into a whole bunch of copies, and then we can go and test those cells for their DNA profile. And then if we have a suspect, we can collect their DNA. But even if we don't, it's possible that person was arrested or basically had their DNA entered into the database. So having that ability has certainly changed crime scene work, investigating crimes of all sorts. But then the technology for even things that aren't possible yet but could be really interesting. We've been able to clone animals now for a little while. Dolly the sheep was the very first animal that was cloned. Now I'm not going to say it's common, but it's a lot more prevalent in livestock. Cloning animals that are the best examples, that have the best marbling in their meat or whatever genetic characteristic you're interested in. Maybe it's an animal that produces fiber, like a goat. Maybe it's a different example in something like bull riding, where bulls are cloned from bulls that were really good bucking bulls. And so whether we're using it, in the agricultural industry, or the idea of people cloning their pets, they don't want to lose them, so they actually have the ability in many cases to be able to clone if they have the money and so forth, to sort of bring their pet back. Now, there's no guarantee that animal's going to have the exact same temperament or personality, but it's certainly something that, like so much of what's going on today, technology-wise, so much that we couldn't even have imagined would be possible not that long ago. So it's also interesting and scary to think about what could be possible in the near future or the distant future. Jurassic Park's, the original movie, came out a long time ago, and it was based on, the premise of being able to produce extinct animals and, extract DNA from, in their case, they used mosquitoes that were fossilized in amber. And of course, if the mosquitoes sucking blood out of a dinosaur, those dinosaur cells are in there. And, if you read the book, there's a lot more detail than what the movie shows about the science behind that. And what I always liked about the author, Michael Crichton, who did Jurassic Park, but he did a bunch of other books, and a lot of them were turned into movies, is that he was a doctor, a medical doctor, and took the science side of his movies seriously, to the point where, again, being a scientist, you're reading the book and going, kind of, I didn't know that was possible, or I wonder if that really is possible, because there's so much of it that's real or realistic or just beyond the edge of what's possible now. We can't take dinosaurs and bring them back yet, or may never be able to. And actually now the technology that scientists are looking at to bring back things like woolly mammoths is actually going in, from what I understand, a little bit of a different direction than what they originally were looking at. But it may be possible to bring back extinct animals, and so then that that brings up a whole lot of moral questions, like there often are in science. just because we can do something, should we, or do we feel obligated to do something because we can? And if an animal went extinct, it was for a lot of reasons, including the place it lived, the habitat's just not suitable anymore. And so if there's no longer suitable habitat, what do you do with these things? Do you clone them? Do you put them in zoos? Well, wait a minute, that's kind of Jurassic Park, right? Build a zoo for these animals, and at least then people can see them, as long as the electric fence doesn't go out, right? So anyway, it's going to be interesting as this DNA technology keeps evolving as to what might be possible in the future. Cloning ourselves when we get old so we don't die, we continue to exist in some form or another. So Watson and Crick are the two scientists that are typically acknowledged for being the individuals that were able to first identify the structure of DNA, basically illustrated in this X-ray, the idea that, sorry here, let me try this again here. The idea that you've got this figure 8 pattern, which if you look at DNA from one perspective, you sort of see this like spiral staircase pattern, and it's hard to maybe picture how that relates to the spiral staircase idea, but they were able to show that it is indeed, DNA is indeed composed of this spiral staircase sort of pattern. So I'm going to show a video in a minute or two here, about 10 minutes long, that kind of goes through the whole process of showing you how DNA looks and what it's made up of, and how, really importantly, how it makes copies of itself. Again, being able to go from one cell and then making copies of our cells, but every cell, of course, has a copy of our DNA, and every cell needs to have that DNA. And so that's kind of a really interesting process. It is a bit complicated, but again, hopefully between reading and studying the imagery and then watching the video, hopefully it'll help clear it up a little bit. So each DNA nucleotide, which is kind of a little subunit of DNA, is made up of 3 basic things. It's made up of a sugar, a deoxyribose sugar, and so at each of these junctions we have a carbon molecule, so the sugar is made up of a carbon backbone, and they're actually numbered for a particular reason that we'll look at in just a minute. The DNA nucleotide is also made up of a phosphate group. Again, at the core of that is a molecule of phosphorus. And then it's also made up of one of 4 possible bases, at least DNA. There are 2 other bases in another molecule, but as far as DNA, there are 4 possible bases, and we refer to them as nitrogenous bases. As you can tell, because we have these nitrogen molecules that are, again, kind of the backbone of the base. And so these 4 bases are made up of a group of molecules called pyrimidines, and the 2 bases that are pyrimidines are cytosine, which we abbreviate with the C, and thymine, which we abbreviate with the T. Now, again, for the purpose of this class, I'm not, going to ask you to memorize any of the chemical formulas. But you should know that a DNA molecule is composed of these three things. The DNA nucleotide is composed of a sugar, a base, and a phosphate group. And then you should also know that those bases can either be pyrimidines, which are cytosine and thymine, or they can be purines, which are guanine and adenine. And again, those are abbreviated G for guanine and A for adenine. So again, understanding the basic structure. So these are the individual components that when you pair them all up, they end up composing the spiral staircase structure that makes up the DNA molecule. Each one of these nucleotides is sort of half of one rung of a ladder. It's half of the rung and half of the upright. So when you have two nucleotides that are matched up, and we'll talk about how they pair up in a minute, you get a full rung and then part of the upright support of what again becomes that spiral staircase that ultimately makes up the DNA molecule. So now we back up a little bit, a little slightly different perspective, and we can kind of draw. Notice the bases are here and then the sugar phosphate backbones. So we kind of draw a line through the middle here. And so one of these would be what we were looking at on the previous slide. The sugar backbone, the phosphate group, and then the nitrogenous base. In this instance, it happens to be guanine. And then its counterpart on the other side, and again, you refer to the bigger DNA model here, that these color-coded rungs or half of the rung of the ladder are the base pairs. So the one I just circled there would be, well, in this case, the one I circled is actually the thymine. Because notice they're color-coded here, red, yellow, blue, and green for adenine, thymine, guanine, and cytosine. But so the base pair is equivalent to the rung of the ladder, and then this structure out here is actually the sugar phosphate backbone part of the molecule. So there's a very specific pattern that's followed here, that guanine and cytosine, those bases always pair up with each other, as do thymine and adenine. Those two bases always pair up with each other. It's sort of like, they fit. Adenine and thymine fit together. Adenine doesn't fit with guanine or cytosine. It only fits with thymine. And so just out of those combinations, adenine and thymine always pairing up, guanine and cytosine always pairing up, then just the pattern of those pairs connecting along with, again, their sugar and phosphate backbone, all of that then combines to form our unique DNA, just depending on the order of these pairs and the repeated number of times that they bond up with each other. Again, pretty amazing to me anyway, how just these four things basically in different orders create all of the diversity of life on this planet. Even just with humans, but with other mammals, other animals, we're only working with these four base pairs, and just the order and the number and all that of these are what makes up the huge amount of diversity of life on the planet. So again, the kind of take-home message is make sure you understand which pairs go together, understand the basic structure of the DNA molecule. Again, that it's made up of sugar and phosphate backbones with their associated base, and that guanine and cytosine are the bases that always pair up. Thymine and adenine are also the bases that always make pair bonds with each other. And notice the molecules at play here, or the type of bond that's at play here, are hydrogen bonds. The hydrogen bonds are what connects the base pairs together. So those bases are connected to each other via the hydrogen bonds. So just a reminder that when we're looking at the individual strands of DNA, again, we have the base pairs here that are connected by hydrogen bonds, and the sugar backbone there that makes up the sort of uprights of this ladder structure. Before the DNA can be replicated, remember, it has to go through that process that we looked at in the previous section, where this long, thin strand of DNA starts wrapping itself around these histones, these proteins. So here we have those proteins, and here is that strand of DNA that is starting to wrap itself around. And so then each of these subunits, which is what we have in the slide above, those repeat wrapping, bundling themselves, or compaction, that ultimately leads to these sort of like almost flower petal patterns, until we eventually get those compacted enough to have this sort of normal shape that we think of as the chromosome. And of course, this is when it's compacted enough that it's visible more easily under a normal light microscope. And so this is what we were observing in the section where we were talking about mitosis and meiosis. So just trying to, again, reminding you the perspective here, that these are big enough to see with a normal light microscope, but as they begin to unravel and we get to that normal sort of double helix structure of the DNA molecule, it's totally unraveled and uncompacted, a very long, thin little strand of information. One of the fundamentally important things about DNA, although life in general, is that it has to be able to make copies of itself, right? We talked about reproduction, sexual reproduction, asexual reproduction. At the whole organism level, humans have to be able to make copies of themselves for humans to continue existing, as does all other life. But then, of course, that also means that at a cellular level, we have to be able to make copies of ourselves, whether it's initially to be able to grow from a single cell, an egg, into the trillions of cells that make up a whole living thing, or whether it's the DNA within those cells that has to be able to copy itself in order that every cell that's made has that same DNA profile. So we're looking at the strands of DNA again here in a slightly different perspective. So again, we have the sort of the two backbones of the DNA molecule, and then the bases, the AT and GC, that always pair up. And notice again, these are just repeated throughout the entire length of the DNA molecule. The thing is that two strands of DNA are complementary, meaning that the bases or the order of the bases on one strand can be used to make an identical copy of the opposite end. So just the picking one side of the DNA strand here. This side or half of the strand has the ability to be able to make the other side of the strand so that it can basically make a completely new strand of DNA that is identical to the other one. And there's a very specific process that it goes through to do that. The video that we'll look at here in just a minute sort of does a good job of explaining what these numbers represent. But notice that they run in opposite directions. From the 5' end towards the 3', from the 5' end towards the 3' end. That number refers to the prime, and there's actually 1, 2, 3, 4, and 5 prime. That refers to the numbering or the order of the carbons on the sugar molecule. And so it's the order basically that that the DNA is replicated. So that's kind of the easy way to think about it. If you're thinking there is such a thing as an easy way to think about DNA replication. So there are a few different models or ways that science believes explains the way the DNA is replicated. So what I want to do is cut to a good little YouTube video here for a few minutes. Again, I think it runs 10 minutes. And I think overall it does a better job than I can do even of explaining or showing how this whole process of DNA replication works. So I'm going to fire that up. Again, it goes for about 10 minutes. And then we'll come back, kind of review what happened there, and go from there. So here we go. a second. As you can see in the video that popped up there, the other couple of topics that are covered in this chapter, again, after DNA replication, are how DNA is repaired, and then transcription and translocation. I'm not going to take the time to run through his video clip on those two topics, but they are in the book. They are part of the content. I probably would, if I were you, you know, go into YouTube and search those topics and maybe take the 10 minutes to watch a video clip. I like this guy. He does a really good job of explaining things. You know, again, probably better than I can even for this topic anyway. So I would really suggest kind of seeking him out, Bosman Science or Bozeman Science, and then watch the sort of the rest of the story about DNA as well, because again, it is part of the process, and I think watching these little you know, 10, 12-minute video clips is probably going to do a lot to help you get a little better understanding. He had also mentioned that the actual sort of computer simulations are a little harder to follow as far as how more realistically DNA replication looks. And again, I'm not going to show it here, but in the slideshow that you're looking at, the link that says 3D animation is actually one of those computer simulations where you can actually see sort of in real time what it actually looks like when your cell is, when DNA in your cell is replicating itself. So again, I think it's worth the 10 minutes to watch his other video and maybe take a few minutes to watch the 3D animation. So the rest of this section just summarizes what he covered, maybe using a little different imagery, but again, it's the same thing that basically is the summary of DNA replication, the helicase unwinding, in other words, kind of unzipping the two strands of DNA, all of the very important enzymes that come into play, the the DNA polymerase that basically is important at the replication fork, primase, the RNA primers that come into play here to rebuild the fragments, DNA ligase that kind of mops up the loose ends, so to speak. So again, just kind of more reminders of how important enzymes are in making everything about us work properly. So I'm not going to kind of go back and explain the process all over again, because again, I think he did a better job than I can. But hopefully between the book, between reviewing the slides, maybe watching the video clip again, or just Googling some other ones that might be out there, it's important to, again, at least understand. You're not going to be asked in this class to be able to like explain the process from beginning to end. But with that said, you still have to kind of understand what's happening. With respect to the next quiz, the one that'll cover this material, there'll be some easy questions. You know, there'll be a list of things and maybe a question like which of the following are not an example of a nitrogenous base. So as long as you know adenine, thymine, guanine, and cytosine, if there's another name listed on there, you'd know which one isn't. But there could be some more slightly more detailed questions about the process of DNA replication. So again, at least get a basic understanding of the process. One other item that I, excuse me, that I do want to spend just a couple of minutes talking about deals with the process of aging. So the end of the chromosomes, called telomeres, are maintained, or maybe you could think of it this way. The end of something, the end of a rope, maybe, for example, you've got a little piece of rope that you use to tie stuff down in the back of your pickup truck. And over time, the end of that rope can kind of get frayed, right? It can start kind of getting frazzled and frayed, the ends, coming unwound a little bit. And the ends of our chromosomes can do the same thing over time. And so we have an enzyme, imagine that helps in trying to keep those ends from fraying. Maybe the equivalent of you using a lighter on the end of a nylon rope to kind of hold it together so it doesn't just keep unraveling. And so the enzyme telomerase helps kind of keep the ends of our chromosomes from fraying or fragmenting. And so where this comes into play is that in our somatic cells, so again, all of our cells except for those in our testes and ovaries, that enzyme telomerase doesn't have an impact. It's not active. It's not activated, so it's not doing anything. And that's the short story of why living things age, why things don't live forever. So the process of that enzyme maintaining the ends of our chromosomes in our sex chromosomes keeps those copies nice and in perfect shape, so that we can make perfect copies of them to be able to push on to the future cells that have nice, clean, crisp copies. But again, our bodies age, and that's literally because our chromosomes are kind of coming unraveled. So this slide shows the process of what telomerase does associated with RNA, and basically it complements the end of the 3' strand, the overhang, and so basically it creates or is used to synthesize the complementary strand. So again, if this is the very end of that strand, we're basically making copies of it, and then the telomerase shifts and repeats. And so that complementary strand is maintained. The end of it is maintained. Whereas again, as we age, our bodies age, and therefore the somatic cells are sort of responsible for that. As they fray and sort of come unraveled, it causes the process we call aging. And it really wasn't until 2009, so not that long ago, that a scientist discovered how telomerase actually works. So we've obviously known for as long as humans have existed that we get old and die. But obviously, we didn't know how that process happened. And certainly, probably there are few things that we care more about than, getting old. And how many products are there on the market that help to claim that they can slow the process of aging or reverse it. The kind of topical skincare products obviously can't do anything about getting old. They can mask the effects, hide wrinkles, that kind of stuff. But there are more products that I've noticed as well that are claiming to actually enhance your body's ability to fight off aging. And a lot of times they claim those have to do with enhancing the repair process, which again basically is affected by the enzyme telomerase. So there may again be a point where we can engineer something at a microscopic level, nanotechnology, that we could make sort of little artificial repairs, or engineer something that we can put in a body that goes and maintains the telomeres, and therefore would have some impact on us aging. Again, we're not there yet. Should we even try to do that? But again, questions that always come up in science. Just because we can do something, does that necessarily mean we always should? And then, of course, along the way, we talked about in interphase of the cell cycle that there are points along the way where there are quality control checkpoints, looking for errors in copying things. And so from the standpoint of the DNA, which remember is copied during interphase, there is, again, surprise, another enzyme, that DNA polymerase, that proofreads, goes along the strand of DNA, making sure that things are correct, that they were copied correctly, and then it finds areas where there are issues and detects that after the DNA strand has been replicated. They actually serve to remove or cut away the part that was copied incorrectly, and then a new strand is synthesized by nuclease action. The gap that was cut away is now filled with the correct base pairs that were incorrectly copied the first time. And then that hopefully avoids there being mutations, spontaneous random changes. Now, keep in mind that basically life on this planet has evolved the way it has very much because of these types of mutations, these sort of random accidental issues that come up when segments of DNA strands are incorrectly copied. Most of the time, these mutations are bad. They create problems that usually results in the death of the organism. But there are occasions, far less frequent, but when these mutations that again have happened by accident, again, think of the billions and billions and billions of times that DNA is copied in your body, or in the process of a cell developing, they're bound to be accidents. And again, most of those mutations are bad, but occasionally they're neutral, they're not good or bad, and then they may just stay there. And then that's something that's in that gene pool, and it may never show up. But there are other instances where these mutations happen and create sort of an instant change. Now remember, just because that mutation is there, and even if it's a positive thing, even if it's something that helps something survive, for example, maybe a random mutation that gives an individual just a little better vision. Maybe they can see colors just a little differently than other individuals, and maybe the ability to see colors a little differently helps them be a better hunter, or helps them be better at hiding or seeing a predator. Just because those mutations are there, and even if they're in the DNA, remember those mutations still have to be transmitted to the next generation. And that's certainly not a given either, as we might remember when we talked about what actually happens during meiosis. So really, the upcoming topic that we're going to talk about, talking about evolution, really is rooted right here on this slide. The fact that these mutations that happen when they're on rare instances something that might be better to help that organism survive, they still have to be transmitted from one generation to the next, to the next, to the next. But if that process happens over millions of years, has resulted in the process that we acknowledge as evolution, the process of living things changing over time, adapting to changes around them, whether those changes are climate or the habitat itself or whatever. So we'll get back to that discussion in an upcoming next chapter. Also, part of this process of aging is that all life on the planet is exposed to ultraviolet radiation, right? There's a certain amount of that that's coming from the sun. And UV, generally speaking, is very harmful to cells. One of the reasons that is that these thiamines, when exposed to UV, the ones that happen to be paired up next to each other can create these things called thymine dimers, which in normal cells, they're identified in the process we just talked briefly about up top. They're cut away and they're replaced. But when they aren't, when they're missed, then they can cause mutations. And again, although most of those mutations tend to be bad, they certainly can be beneficial at times, ultimately, which is good for living things to be able to evolve and adapt over time. Again, I mentioned the guy that we were just watching had that other video, 10-minute video on transcription and translation. And so I would really suggest watching that. But again, I'm not gonna worry about going into any more detail with that. But it's still a part of the entire process of being able to replicate your DNA. Have a little bit of content in the book about RNA and the different types of RNAs. RNA, transfer RNA kind of is a translator. It kind of translates RNA language into proteins. So basically building proteins and ultimately then chains of proteins into amino acids. And again, looking at an image of this or reading the words in the book are probably not going to do it as much justice as being able to watch the videos as well. So I would really, again, recommend that you do that. You Google YouTube and look for the Bozeman guy there. And so a lot of this has to do with synthesizing proteins, like I talked about. But RNA has such an important role as well that you need to at least understand, again, in pretty basic terms, what it does. So then your book has a table here, kind of, that talks about how transcription generates a copy of genes with our, ACGU. Notice it's not A, C, G, and T. Thymine is replaced by uracil, and in RNA, so you have one of those base pairs that's different in the RNA. And then translation of the messenger RNA, the template that messenger RNA builds to convert information into the proteins that make up the different amino acids. Again, just sequences, these 3 nucleotide sequences that make up all of the 20 different amino acids. So again, there's a whole nother side to making copies of things. It's not strictly about copying the DNA molecules. Remember, when a cell makes copies of itself, yes, the DNA is a pretty important part of that. but it has to make copies of everything that's in the cell, including the proteins, for example, and make all of those copies identical, barring errors for the daughter cells. All right, so again, I know I kind of glossed over those last couple of topics. They are covered in the book. I also just don't want you to get too bogged down in some of the other content here. It is important. Again, it's kind of fundamental to knowing who we are or what life is on this planet. But I also know that for students who aren't science majors, it starts getting just like a bunch of blah, 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 you know, Charlie Brown listening to his parents talk or his teacher talk. And again, it's important, and if you're interested, I would really recommend that you spend some time really, you know, studying it and reading more about it. And again, there's so many good videos and animations out there that have the technology to show you stuff that I can't generate myself. And oftentimes, you know, the combination of listening and reading and watching videos, especially today with the technology we have to make these videos, it really helps you understand things better. So pay attention to the key terms. That's going to be important for the next quiz with respect to this content. And again, have a basic understanding of what makes up DNA, different components, and a basic understanding of how it actually is able to make copies of itself. And then at least in general terms, what transcription and translation are. And if you have that sort of you know, ready to go for the quiz in your knowledge bank, then you should do all right on this content. And again, so now after this point, we're moving into the bigger stuff. It's not focusing on the stuff that we can't see, although that's super important. It's getting into the bigger picture stuff. And we'll again be moving on to the topic of evolution in chapter 11 next time.